Hello, beautiful souls, welcome to this quiet space, a place where your mind can slow down and your heart can rest. Before we begin, if this meditation brings you peace, please subscribe and gently tap the like button. Your support helps us continue creating these calm spaces for you. Now, there is nothing you need to do, nothing to solve. Just close your eyes. Take a slow, gentle breath in, and softly breathe out. Let your shoulders relax. Let your body grow heavy. And together, let's begin. Let us begin with the thing most easily misunderstood. When we say the word mushroom, we summon a very specific image, a cap on a stem, perhaps with gills beneath, perhaps growing in a fairy ring in the morning grass, perhaps arranged prettily on a kitchen counter. The mushroom in our imagination is the whole thing. It is the organism. It is, as far as most of us instinctively think, the fungus. But the mushroom, strictly speaking, is only a body part. A temporary one, at that. To understand what a fungus truly is, we need to follow it downward, past the stem's base, past the point where it meets the soil, down through the compressed dark of the earth, and there, spreading outward in every direction like threads of pale silk woven into the grain of the soil itself, we find the real body of the organism. This is the mycelium. And compared to the mushroom above it, the mycelium is something else entirely. Mycelium is composed of individual threads called hyphae, filaments so fine that a single one would be invisible to the naked eye, thinner than a human hair, sometimes thinner still. Each hypha is essentially a long, branching tube, its walls made of a tough but flexible material, its interior filled with living cytoplasm that streams and flows as the hyphal tip extends outward. Hyphae branch and rebranch with extraordinary regularity, splitting apart to explore new territory, fusing where they meet compatible strands from neighboring networks, weaving themselves into a three-dimensional structure of almost incomprehensible complexity. The result is something that resists easy comparison, though it is tempting to reach for familiar metaphors. You might imagine it as a net, but that is too regular, too orderly. You might imagine it as a tangle, but that undersells its purposeful architecture. Perhaps it is closer to a constellation drawn not among stars but among soil particles and root hairs and fragments of decaying wood, a vast, branching diagram of connection, spreading through space not according to a predetermined plan but in constant, adaptive response to the chemistry and texture of the world through which it grows. This mycelium is the main body of the fungal organism. The part that feeds. The part that breathes, after a fashion. The part that perceives its environment through chemical gradients and responds accordingly. The part that may persist, in some cases, for decades or centuries without pausing. In certain remarkable cases, which we will visit later, mycelial networks have been found covering areas of many acres, representing a single living organism of extraordinary age. The mushroom, then, is what mycologists call the fruiting body. It is produced by the mycelium at particular moments, typically when environmental conditions align in just the right way. Moisture and temperature and season and the chemistry of the surrounding soil all play their parts in triggering the mycelium below to push its energy into producing this brief, reproductive structure above. There is a moment, just before dawn, when the forest holds its breath. The light has not yet arrived. The mist has settled low against the ground, drifting in slow, unhurried columns between the trunks of old trees. Somewhere deep within the wood, a single drop of water gathers at the edge of a leaf, swells slowly in the cool air, and falls, tumbling downward through the layered dark until it disappears into the soil below. And then, almost imperceptibly, something begins to move. Not the kind of movement you would notice if you were walking through. Not the rustle of an animal or the creak of a branch in wind. This is a quieter motion entirely, a slow unfolding at the boundary between the seen and the unseen. From beneath the pressed-down mat of last autumn's leaves, something pale and soft is emerging. A cap, rounded and smooth, no wider than a thumbnail. Then another beside it. Then a cluster, rising gently from the earth like small pale moons surfacing through still water. By the time the first light reaches the forest floor, they are already there. Mushrooms. Dozens of them, gathered in the hollows between roots, pushing upward through the damp decay of fallen bark, appearing along the curved lip of a rotting log as though they had always been waiting, patient and quiet, for exactly this moment to arrive. Most people, walking through that forest in the morning hours, would pause. They might crouch down, study the delicate gills beneath a cap, notice the way the stem emerges cleanly from the leaf litter, and then stand again, moving on with only a vague impression of having seen something lovely and strange. Mushrooms, after all, seem like visitors. Brief, soft-bodied, oddly beautiful things that come and go with the rain. But here's what those morning walkers rarely consider. The mushroom they are looking at is not where the organism begins. It is not, in any meaningful sense, the creature itself. It is the outermost expression of something vast, a brief, visible flourish produced by a network so intricate, so ancient, so pervasive beneath the ground they are standing on, that to call it hidden seems almost too small a word. What if the most important life on Earth is the one we rarely see? This is not a question designed to startle you. It is simply an invitation to slow down, to look beneath the surface of the familiar, and to consider that what we understand as nature, as the living world, as the deep interconnected web of things that grow and breathe and decay, may be shaped in ways we are only beginning to understand by organisms that conduct their entire existence underground, in darkness, in silence, and across timescales that dwarf our own. Fungi, the kingdom to which mushrooms belong, are neither plants nor animals. They are something older, something stranger, something that has been quietly working at the foundations of terrestrial life since long before the first forests existed. They are decomposers and symbionts. They are architects of soil and partners of trees. They have colonized the driest deserts, the deepest ocean floors, the frozen edges of polar ice. They produce the enzymes that break stone to mineral and wood to earth. They are woven, invisibly, into the fabric of nearly every ecosystem on land. And yet, for most of human history, and still, for most people today, fungi are understood primarily through their fruits, those brief, soft caps that appear after rain and vanish before the week is done. The mushroom, it turns out, is only the visible whisper of a vast hidden kingdom. Come, then, let us go downward, beneath the leaf litter, beneath the compressed dark of the topsoil, into the slow, cool, intricate world where most of life's real work is done, quietly, patiently, without applause, and without end. This is an invitation into stillness, into the kind of attention that requires us to move slowly and look carefully, to sit with the not immediately obvious, to allow time to do what time does best, reveal. The forest is breathing below. It has been breathing for hundreds of millions of years. And tonight, beneath your feet, beneath every field and garden and ancient wood on this planet, it is breathing still. The word fruiting is useful here, because it draws a connection to something more familiar. In the same way that a tree, the great, long-lived, rooted, perennial organism, produces fruit, those temporary, sugar-rich structures designed to carry seeds outward into the world, to disperse the genetic material of the organism to new ground, so too does the fungal mycelium produce mushrooms. The tree is not the apple. The mycelium is not the mushroom. The apple is a brief, purposeful structure. So is the mushroom. The difference, of course, is that we rarely make the mistake of thinking a tree is an apple. But with fungi, this conflation happens constantly, and understandably so, because the mycelium beneath the ground is invisible to us while the mushroom above is what we see. This has consequences for how we understand the lifespan of fungal organisms. When a mushroom withers and collapses after a few days in the sun, when it turns soft and dark and seems to dissolve back into the earth, we might assume the organism is gone. But the mycelium from which it emerged is still there, still growing, still exploring, still exchanging materials with the roots of nearby plants, still digesting the cellulose in that old log, still doing the slow, essential work that fungi do. The mushroom was only a message sent upward into the visible world. The organism that sent it remains below. This realization quietly shifts something in the way a forest looks. Those clusters of mushrooms at the base of an oak tree, are they separate individuals, or are they all expressions of the same underground body? In many cases, research suggests the latter. What appears on the surface as a dozen distinct mushrooms may all be the fruiting bodies of a single mycelial network threading its way through the root zone of several trees at once. Hyphae, as they grow, do something rather elegant. They extend at their tips, always at their tips, pushing outward into the substrate around them by secreting enzymes ahead of their advance. These enzymes break down the complex molecules of whatever they encounter: wood, leaf litter, the coatings of soil particles, and the resulting simpler compounds are absorbed back into the hypha through its wall. This is how fungi feed, externally, chemically, in a process that is almost the opposite of how animals eat. Animals take things inside and digest them internally. Fungi, in a sense, digest things outside themselves and then absorb the products of that digestion. As hyphae branch and grow and fuse and spread, the mycelium develops what might be thought of as channels, pathways along which materials can travel back and forth through the network. Nutrients gathered at one end can be transported toward areas of active growth at the other. Water absorbed in one part of the network can move toward regions where the soil is drier. This capacity for internal transport gives the mycelium a kind of distributed functionality that goes well beyond what you might expect from something so small and so seemingly simple at the level of the individual hypha. The mushroom, when it finally forms, draws heavily on this internal transport system. Materials are moved toward the developing fruiting body in large quantities. Water, in particular, plays a dramatic role. It is the rapid uptake of water that allows a mushroom to expand with such surprising speed, sometimes going from a tiny rounded primordium to a fully expanded cap in the space of a few hours. That almost magical overnight appearance of mushrooms after rain is, in part, a story of hydraulics, water flooding upward into waiting structures, inflating them from within, unfolding their caps, extending their stems with a quiet urgency that needs no muscles and no bones. And then, once the spores have been released, once the mushroom has done its reproductive work, it softens, collapses, returns to the earth from which it came. The mycelium continues. The story below the ground goes on. For a very long time, scientists placed fungi among the plants. This seemed reasonable enough. Fungi did not move. They grew from the ground. They were rooted, in a manner of speaking, to their substrates. They were not warm-blooded. They did not hunt. And so, for centuries, the taxonomy of living things placed them alongside ferns and mosses and flowering plants, in the broad and comfortable category of vegetation. It was not until the 20th century, with the development of more sophisticated tools for understanding the biochemical and genetic relationships between organisms, that the full strangeness of fungi began to emerge. Fungi, it turned out, were not merely atypical plants. They were not plants at all. They belonged to an entirely separate kingdom of life, one with its own ancient lineage, its own distinct cellular architecture, its own peculiar relationship with the world, and its own evolutionary history stretching back over 1 billion years. The differences, once you know where to look, are everywhere. Begin with the cell wall. Plants build their cell walls from cellulose, long chains of glucose linked together in a particular configuration that gives plant cells their characteristic firmness. Animal cells have no cell walls at all. But fungal cell walls are made primarily from chitin, the same tough, flexible material that forms the outer shells of insects and crustaceans. A mushroom cap and a crab shell share, at the molecular level, the same structural ingredient. This is not a coincidence. It is a signal of deep evolutionary kinship. The implications go further. Chitin-based cell walls confer a different set of properties than cellulose-based ones. They are more flexible, more resistant to certain environmental stresses. And the fact that fungi share this molecular building block with arthropods rather than with plants tells us something important about where fungi sit in the great tree of life. Genetically, fungi are more closely related to animals than they are to plants. They branched from the lineage that would eventually give rise to animals at some point deep in evolutionary time, long before the first multicellular creatures emerged from the oceans to colonize dry land. This relationship is reflected in how fungi handle nitrogen, an element essential to building proteins. Plants obtain nitrogen from the soil through their roots, absorbing simple mineral forms of it. Animals obtain nitrogen by eating other organisms. Fungi, like animals, store their energy not as starch but as glycogen. Like animals, they cannot photosynthesize. They cannot capture the energy of sunlight and use it to build sugars from carbon dioxide. They must obtain their energy and their nitrogen from organic sources. They must consume. But here's where fungi diverge from animals so completely that the divergence requires its own category of understanding. Fungi do not eat by ingesting. They eat by excreting. The hyphal tip of a growing fungus is, in a very real sense, a chemical weapon directed outward. It secretes enzymes, proteins that act as molecular scissors, cutting apart the complex chemical bonds that hold together the polymers of wood, leaf, and other organic material. Lignin, the extraordinarily tough substance that gives wood its structural strength, is broken open by enzymes that few other organisms on Earth can produce. Cellulose, the primary component of plant cell walls, is cleaved apart by cellulases. Complex proteins are denatured by proteases. Long-chain molecules are unwound, fragmented, dissolved. The resulting simpler compounds, sugars, amino acids, mineral ions, are then absorbed through the fungal cell wall. The fungus has, in effect, digested its meal before touching it. The external environment becomes, in some sense, an extension of the fungal body, a zone of active chemical processing that exists in the space around the hyphal tips. This mode of nutrition, called absorptive heterotrophy, places fungi in a category entirely their own. They are consumers, yes. They depend on organic material produced by other organisms. But the mechanism by which they consume is so different from that of any animal that to compare them seems almost misleading. Fungi also reproduce in ways that reflect their separateness. They produce spores, reproductive cells that come in an extraordinary diversity of forms. Some fungal spores are sexual, produced through the fusion of compatible hyphae and carrying recombined genetic material. Some are asexual, produced by the mycelium simply budding off copies of itself. Some are designed to survive drought. Some are designed to survive heat. Some are coated in water-repellent compounds that allow them to float on the surface tension of water. Some are equipped with hooks and sticky surfaces that allow them to attach to passing insects or the fur of mammals. The life cycle of many fungi involves multiple stages, sometimes with completely different forms at each stage, and the genetic machinery governing these transitions is complex and, in many species, still only partially understood. What this all amounts to is a portrait of life organized along completely different principles than either plants or animals. If you imagine the history of life on Earth as a vast experiment in the chemistry of existence, an ongoing trial of different strategies for capturing energy, reproducing, responding to the environment, and persisting through time, then fungi represent one of the most successful and distinctive outcomes of that experiment. Not better than plants. Not worse than animals. Simply different. A parallel answer to the same fundamental questions. A kingdom of their own. And what a kingdom it is. Scientists have so far described approximately 150,000 species of fungi, but estimates of the true number of fungal species on Earth range from 2 million to perhaps 10 million or more, which suggests that the vast majority of fungal life remains unstudied, unnamed, and unknown. In every soil sample taken from anywhere on Earth, there are fungal organisms. In the bark of trees, in the guts of insects, in the deep sediments of the ocean floor, on the surfaces of rocks in Antarctica, in the water that falls as rain, fungi are present everywhere, doing what they have always done, quietly and without interruption, for longer than there have been forests, longer than there have been fish, longer, perhaps, than there has been any complex life at all. They predate our wonder by an almost incomprehensible margin. And they will, in all likelihood, long outlast it. Imagine, for a moment, a forest without decay. Picture it clearly. The trees still grow, the seasons still turn, the leaves still fall each autumn in their slow golden cascades. But nothing rots. The fallen leaves pile up, year after year, decade after decade, building into thick compressed mats that grow ever deeper. The fallen trees remain, hard, intact, their trunks strewn across the forest floor like the ruins of buildings no one has cleared away. The dead roots stay locked in the soil. The bodies of animals, birds, insects, deer, remain wherever they fall, perfectly preserved, undisturbed by the processes that would normally claim them. In this imaginary forest without decay, the surface of the earth would eventually become unnavigable. Buried beneath millennia of accumulated organic matter, the soil itself would be inaccessible to new roots. The minerals bound up in all that undegraded wood and leaf and bone would be unreachable, locked away from the living organisms that need them. The carbon contained in all that unrotted matter would remain sequestered in dead tissue, unavailable for recycling back into the atmosphere or into new living cells. Life, as we know it on land, would grind slowly toward its own stagnation. This scenario remains, thankfully, entirely hypothetical, because fungi exist. Of all the roles that fungi play in the living world, and they play many, perhaps none is more foundational, more quietly essential, more invisible to casual appreciation, than their role as decomposers. Saprotrophic fungi, as mycologists call this group, are the organisms primarily responsible for breaking down the complex organic molecules in dead plant material and returning their component elements to the soil, the water, and the air. Let us follow a single fallen tree to understand what this means. A large oak, after perhaps 200 years, finally succumbs. Maybe it is a storm that brings it down, a winter wind that finds the weak point in the root system and leverages the full weight of the canopy until something gives. The tree falls with a sound that echoes through the forest. The canopy above is now open, a shaft of light reaching the forest floor for the first time in decades. And the tree lies still. Within days, sometimes within hours, fungal spores that have been waiting in the leaf litter around the fallen trunk begin to germinate. Hyphae push outward from these germination points, probing the surface of the bark, finding the cracks and gaps where moisture has already begun to collect. Slowly, steadily, the mycelium begins to thread its way into the wood. What it encounters there is a fortress of complexity. Wood is composed primarily of two substances, cellulose, which forms the structural scaffolding of the cell walls, and lignin, which is woven between and around the cellulose fibers like a biological concrete, binding the structure together and conferring extraordinary resistance to mechanical damage and biological attack. Lignin is, in a sense, the reason trees can grow tall. It is one of the most structurally sophisticated and chemically resistant biopolymers that life has ever evolved. And for most organisms, it is virtually impenetrable. For certain fungi, however, particularly the white rot fungi, so named for the pale, bleached appearance they give to degraded wood, lignin is not a barrier but a food source. These organisms produce a suite of specialized enzymes, including peroxidases and lactases, that generate highly reactive chemical species capable of opening the tangled chemical structure of lignin. It is a remarkable feat of biochemistry. Scientists have been studying these enzymatic processes for decades, and the precise mechanisms are still being worked out. What is clear is that the white rot fungi are, essentially, the only organisms on Earth that can efficiently and completely break down lignin under natural conditions. As the mycelium spreads through the wood, the structure of the tree begins to change. The dense, interlocked matrix of cellulose and lignin is gradually broken open. The cellulose, now exposed, is attacked by cellulases, enzymes that snip the long glucose chains into shorter fragments and ultimately into individual sugar molecules that can be absorbed through the hyphal wall. The lignin is progressively oxidized and dissolved, its complex ring structures opened and its carbon atoms made available for the fungal metabolism. From the outside, this process is visible as a slow softening of the wood. What was once a hard, resonant surface becomes first slightly yielding, then crumbly, then porous, then, over years and decades, something that more resembles soil than timber. The mycelium is, quite literally, turning solid wood into earth. But the fungus is not working alone. As it breaks down the wood, it creates habitat and food sources for a vast community of other organisms. Bacteria colonize the decomposing tissue, processing compounds that the fungus has released. Springtails and mites and millipedes move in to feed on the mycelium and the softened wood alike. Beetles bore through the structure, their larvae feeding on both the wood and the fungi growing within it. The fallen tree becomes, over time, a layered community of organisms, a whole ecosystem within an ecosystem, its structure gradually dissolving into the surrounding soil. And as it dissolves, it returns. The carbon that was locked in the wood cellulose and lignin is released, partly into the atmosphere as carbon dioxide through the respiration of all the organisms feeding on the log, and partly into the soil in the form of stable organic matter. The nitrogen, the phosphorus, the calcium, the potassium, all the mineral nutrients that the tree spent 200 years gathering from the soil around it, are gradually liberated from the complex molecules in which they were stored and returned to forms that plants can absorb through their roots. This is nutrient cycling. And it is, in its quiet way, the process that makes the continuation of terrestrial life possible. Without it, forests would indeed suffocate in their own accumulated debris. Without it, the minerals necessary for plant growth would accumulate in dead tissue and become inaccessible to living organisms. Without it, the soil itself, that remarkable living matrix in which terrestrial ecosystems are rooted, would gradually lose its fertility. Brown rot fungi contribute to this cycling differently than white rot fungi. Rather than breaking down lignin, brown rot fungi focus primarily on cellulose, leaving the lignin largely intact. This produces the crumbling, reddish-brown residue sometimes called red-rotted wood, and the lignin-rich remains left behind contribute to the dark, stable organic matter of forest soils, matter that can persist in the soil for centuries, storing carbon and providing a long-lasting foundation for soil structure and fertility. There are also soft rot fungi, which specialize in attacking the cellulose of wood in moist conditions, producing a characteristic soft, cuboid cracking pattern in degraded timber. And there are fungi that specialize in the decomposition of other materials entirely: dead leaves, the keratin of feathers and horn, the chitin of insect exoskeletons, the complex molecules of animal dung. Each of these decomposer communities, working at different rates and on different substrates, contributes to the vast, continuous cycling of materials through the living world. It is a process without center, and without end. Atoms that today are part of a mushroom cap may, within weeks, be part of a tree root or a beetle's body or a handful of forest soil. The cycling never stops. It only passes through different forms, different organisms, different moments of transformation. And at every stage of that transformation, there is almost certainly a fungus involved. Beneath every healthy forest, a negotiation is constantly in progress. It is not a negotiation conducted in words, of course, or in any medium we could directly perceive. It takes place in the microscopic zone where the tips of plant roots and the tips of fungal hyphae come into contact, a zone so small that you would need a microscope to see it clearly, and so pervasive that in any handful of forest soil, it is occurring thousands of times over. This negotiation is called mycorrhiza, from the Greek for fungus and root, and it represents one of the most widespread and consequential biological partnerships on Earth. To understand what is being exchanged, we need to understand something about the limitations of trees. Trees are extraordinary organisms in many respects. They can grow to tremendous heights. They can live for centuries or millennia. They can pump water from deep underground to their uppermost leaves. They can produce an astonishing array of chemical compounds. But there is one thing they cannot easily do on their own: efficiently gather phosphorus and nitrogen from the soil. Phosphorus, in particular, moves through soil very slowly. It does not dissolve readily in water and does not migrate toward roots the way that some other nutrients do. This means that the zone immediately around a root tip, called the rhizosphere, tends to become depleted in phosphorus relatively quickly, as the root absorbs what is available. To access more phosphorus, the plant must either grow new roots into fresh soil, or find some other means of extending its reach. This is where the fungus comes in. Mycorrhizal fungi, there are many different groups of them, with somewhat different strategies and anatomies, form intimate associations with plant roots. In the most common type, known as arbuscular mycorrhiza, fungal hyphae actually penetrate the cells of the root, forming elaborate branch structures inside the root cells called arbuscules. These are not parasitic invasions. The root cell's membrane remains intact, forming a kind of interface, an envelope around the arbuscule, across which materials can be exchanged between the two organisms. It is an arrangement so intimate that distinguishing where the fungus ends and the plant begins becomes genuinely difficult. In another common form, called ectomycorrhiza, the fungal hyphae do not penetrate the root cells but instead form a dense sheath around the outside of the root and grow inward between the root cells, creating a structure called the Hartig net. Many of the trees in temperate forests, oaks, beeches, pines, birches, form ectomycorrhizal associations, and the fungi involved include many of the most familiar and beloved culinary mushrooms, porcini, chanterelles, truffles, fly agarics. What passes between these two partners? From the plant side, carbon, in the form of sugars. Plants, through photosynthesis, are extraordinarily good at capturing the energy of sunlight and using it to build sugar molecules from carbon dioxide and water. But sugars themselves are not always the limiting factor in plant growth. The limiting factor is often phosphorus and nitrogen, the mineral nutrients needed to build the proteins and nucleic acids that all living things require for growth and reproduction. Evidence suggests that mycorrhizal plants channel a significant portion, in some cases, a very large portion, of the sugars they produce through photosynthesis toward their fungal partners. From the fungal side, phosphorus, nitrogen, and other mineral nutrients, gathered from the soil by the far-reaching hyphal network and delivered to the root interface. The mycelium of a mycorrhizal fungus can extend far beyond the zone immediately around the root, exploring soil that the roots themselves would never reach, accessing pockets of mineral-rich material in the interstices between soil particles, and delivering their chemical contents back toward the root. The mathematics of this exchange are remarkable. A single plant root has a limited surface area available for nutrient absorption. A mycorrhizal fungal network, extending outward from that same root through many meters of soil, can increase the effective absorptive surface available to the plant by a factor of many times over. Some researchers suggest the increase can be an order of magnitude or more, depending on the species involved and the soil conditions. It is worth being careful here about how we characterize this arrangement, because popular accounts have sometimes pushed the description further than the science currently supports. Calling this exchange a tree's choice or describing the network as a deliberate communication system overstates what we can currently demonstrate. What research does show clearly is that the chemical exchange is real, that plants with mycorrhizal partners tend to grow more effectively under nutrient-limited conditions than those without, and that the fungi benefit metabolically from the sugars they receive from plant hosts. What is also becoming clearer, though this remains an area of active research, is that mycelial networks may sometimes connect multiple plant individuals in the same community. When fungal hyphae link the roots of two or more trees, there is at least the possibility that materials might move through the fungal network from one plant to another, along gradients of concentration. Research suggests this occurs in some forest systems. How significant it is as a pathway for resource transfer between trees, and whether it is better understood as a byproduct of the fungal network structure rather than a purposeful function, is still being investigated with great care. What seems less in doubt is that mycorrhizal associations are extraordinarily common. Studies suggest that the majority of land plant species, including most crops and nearly all forest trees, form mycorrhizal associations of one kind or another. These associations appear to have played a role in the initial colonization of land by plants, hundreds of millions of years ago, when early plant ancestors partnered with fungal organisms to cope with the challenges of dry, nutrient-poor terrestrial environments. A tree growing in healthy forest soil, then, is almost certainly not growing alone. Its roots are almost certainly entwined, in the intimate chemical sense of the word, with one or more fungal species that extend its effective reach, that supplement its access to limiting nutrients, and that are in turn sustained by the photosynthetic products of the tree's own leaves. The roots and the fungi are woven together in the most literal sense. They've been woven together, in various forms and with various partners, for perhaps 400 million years. The partnership is older than the forest itself. In the space through which you are breathing right now, there are almost certainly fungal spores. This is not a cause for alarm. It is simply the nature of air on Earth, rich, complex, alive in ways that are mostly invisible to us. Spores are everywhere, above cities and above oceans, above deserts and above rainforests, at ground level and in the upper atmosphere, drifting in currents that carry them across continents and between hemispheres. They settle on every surface. They enter every breath. They are, in a very real sense, the medium through which fungi seed the living world with their possibilities. A single mushroom, during its brief productive life, may release an extraordinary number of spores. The specific numbers vary enormously between species, from millions to billions, and in some cases, estimates extend further still. These spores are released from the gill surfaces beneath the mushroom cap, falling downward through the gap between the gills in a continuous, invisible rain that, in certain species under certain lighting conditions, can actually be seen as a faint drift of powder below the cap. The mechanism by which spores are released varies by species. Many basidiomycetes, the group that includes most familiar mushrooms, use a remarkably elegant mechanism. Each spore is attached to a small structure called a sterigma, and at the moment of release, a tiny drop of water condenses at the base of the spore. The surface tension of this drop changes the balance of forces at the attachment point, and the spore is catapulted, launched with a brief, sharp acceleration, into the air gap between adjacent gill surfaces. The acceleration involved is, relative to the size of the spore, extraordinarily high. This launching mechanism, sometimes called the Buller's drop mechanism after the mycologist who first described it in detail, operates continuously and silently in every mature mushroom, producing the steady drift of spores that disperses the fungus into the surrounding world. Once airborne, a spore's fate depends on chance, on aerodynamics, on the condition of the air through which it travels. Most spores do not travel far. They settle within a short distance of the parent mushroom, where conditions may or may not be favorable for germination. The vast majority of spores will never germinate at all, landing on unsuitable substrates, being consumed by other organisms, or simply failing to encounter the conditions they require. The production of enormous numbers of spores is, in evolutionary terms, a hedge against this overwhelming probability of failure. But some spores do travel. Air currents, the large-scale movements of air driven by thermal differences and pressure gradients, can carry spores upward into higher atmospheric layers where they may travel thousands of kilometers before descending again. Fungal spores have been collected at altitudes of many kilometers. They have been found in the snow of remote mountain ranges far from any known source. They appear in the dust that Saharan winds carry westward across the Atlantic and deposit on the forests of South America. Water is another dispersal medium. Spores that land on the surface of a stream or river may be carried downstream, sometimes adhering to the legs and bodies of aquatic insects that eventually emerge and fly. Spores embedded in soil can be transported during heavy rains when soil particles become suspended in runoff. Fungal material has been found in the sediments of lake beds and ocean floors, deposited from above by the slow settling of airborne particles. Animals, too, serve as spore carriers. Many mushrooms produce volatile aromatic compounds that attract insects. Flies, in particular, are drawn to certain mushroom species and may transport spores on or within their bodies as they move between plants. Some fungi have evolved elaborate relationships with specific insect partners, truffles, which fruit underground and have no need for aerial spore dispersal, rely instead on animals that root for them, boars, deer, squirrels, and of course trained dogs, to dig them up and consume them, releasing their spores in the process of digestion and eventually depositing them in new locations through feces. The spore itself is a remarkable structure, a package containing the essential genetic information of the organism, protected by a wall that must be tough enough to survive the rigors of dispersal while also being capable of breaking open at the right moment to allow germination. Spore walls are often made of spore pollenin, one of the most chemically resistant biological compounds known, capable of surviving extreme temperatures, desiccation, and exposure to ultraviolet radiation. This resistance is why fungal spores can remain viable, capable of germination, for extended periods of time, sometimes years, in conditions that would destroy most biological material. Dormancy is a crucial strategy. A spore that germinates immediately upon landing in an unsuitable location is a spore that has wasted its chance. Many spores remain dormant until specific conditions are met: the presence of moisture, a particular temperature range, certain chemical signals from the surrounding environment. Some spores require a period of cold, a winter's duration, before they will germinate. Others may lie dormant in soil for years, waiting for conditions that may never arrive. The capacity to wait, to persist through unfavorable periods until the moment is right, is one of the deep strategies of fungal life. When conditions are favorable and a spore does germinate, a germ tube emerges from its surface, a single hypha, extending outward into the surrounding medium. This is the beginning of a new mycelium. If the spore lands where conditions are suitable, if there is moisture and appropriate organic material and the chemistry of the soil falls within acceptable parameters, then this single germ tube will branch, and branch again, and over time develop into a new fungal network extending outward in all directions from the point of landing. Most of these new mycelia will grow quietly for some time before producing a fruiting body of their own. Some may grow for years without ever producing a mushroom. Some may merge with compatible mycelia from other spores of the same species, forming a single, larger network. And eventually, when conditions align, some will produce those brief, soft, visible expressions of themselves, caps rising through the leaf litter in the early morning, and release their own clouds of spores into the waiting air. The cycle is ancient and self-renewing. The air is never empty of it. There is a question that emerges. When you spend enough time thinking about mycelial networks, how does a fungus decide where to grow? The question seems, at first glance, trivially simple. A fungus is not a complex animal. It has no brain, no nervous system, no sensory organs in any conventional sense. It cannot see, cannot hear, cannot smell in the way that we understand smelling. And yet, the mycelium of a fungal network does not grow randomly. It explores its environment with what appears, to any careful observer, to be something like purposefulness. It extends toward food sources. It retreats from toxic compounds. It allocates its resources with what seems like efficiency. It builds connections that solve, over time, the geometric problem of how to link many points in a network while minimizing the total length of connections required. This last observation became famous through a remarkable series of experiments conducted in the early years of the 21st century, in which researchers offered the mycelium of a wood decay fungus, a slime mold-like organism, not technically a fungus but often studied alongside them, nutrients arranged to mimic the pattern of cities around Tokyo. They offered it the same nutrients as a kind of map of the region, and they observed, over hours and days, as the organism built a network of connections between those nutrient sources that closely resembled the actual railway networks surrounding Tokyo, a system that human engineers had developed over many decades at great expense. Whether similar experiments with true fungi yield comparable results is an area of ongoing research. What has been demonstrated clearly is that mycelial networks do reorganize in response to resources and obstacles, and that this reorganization has structural properties that researchers describe as efficient. The network avoids dead ends. It creates redundant connections, alternative pathways that allow materials to continue moving even if one route is blocked. It concentrates growth where conditions are favorable and abandons areas that are nutrient poor. How does this happen without a brain? The answer appears to lie in the chemistry of the mycelium itself. Hyphae do not grow in a vacuum. They grow through a chemical landscape, a substrate rich with molecular signals that the growing hyphal tip can detect and respond to. Nutrient-rich areas produce gradients of diffusing chemical compounds that attract hyphal growth. Toxic compounds repel it. The moisture content of the substrate affects the rate of hyphal extension. The mechanical resistance of the material through which the hypha is growing affects how it branches. Each individual hyphal tip, in responding to its immediate chemical environment, contributes to the behavior of the whole network. The network, as a result, behaves as though it has solved a problem, though no single part of it has processed anything resembling a calculation. The solution emerges from the interaction of many simple local responses, each one chemically mediated, each one influencing the gradients experienced by neighboring hyphae. This is sometimes described as distributed computation, or as emergent problem solving. Neither phrase quite captures the strangeness of it. The network's behavior has properties that seem to exceed what any individual part could achieve, and yet those properties arise from exactly those individual parts, without any central coordination, without any privileged node that receives all the information and issues all the commands. There is also evidence that information of a kind can propagate through mycelial networks. Chemical signals released at one point in the network, triggered, for example, by injury or by the detection of a food source, may influence the behavior of hyphae elsewhere in the same network. How far such signals propagate, and how reliably they alter behavior at a distance, varies by species and context and is still being studied. But the observation that a mycelial network can, in at least some circumstances, communicate something from one part of itself to another, without a nervous system and without electrical impulses, is genuinely intriguing. It would be a mistake to read into this any implication of consciousness. There is no evidence that fungi experience anything, that they have preferences in any subjective sense, or that the chemical processes within their hyphae correspond to anything like awareness or intention. The word intelligence, as applied to fungi, requires constant qualification. What we are observing is not a brain-like process relocated to a different substrate. It is something categorically different, a form of biological responsiveness that achieves some of the same functional outcomes as intelligence, without, as far as we can tell, involving anything resembling the neural architecture through which intelligence operates in animals. And perhaps that is what makes it most interesting. It suggests that the capacity to solve spatial problems, to allocate resources efficiently, to respond adaptively to a complex and changing environment, these capacities are not the exclusive property of nervous systems. They can arise from much simpler building blocks, given the right arrangement and the right chemical medium. The mycelium is not thinking, but it is doing something that, from a sufficient distance, can look rather like thinking. And that observation opens up questions about the nature of problem solving, the relationship between structure and function, and the many different ways that life on Earth has developed the capacity to respond to the world around it, questions that scientists, philosophers, and curious observers are still working to answer. There is also something worth noting about the adaptive capacity of fungal networks over time. When environmental conditions change, when a food source is exhausted, when a portion of the network is damaged by a passing animal, when temperature or moisture shifts, the mycelium reorganizes. Growth patterns shift. Resources are reallocated. New hyphae extend in different directions. The network, without any plan and without any planner, adjusts itself to new circumstances. This capacity for self-reorganization, operating continuously and without rest, is part of what makes the mycelium such an effective organism. It does not commit irreversibly to any single pathway. It spreads options. It maintains flexibility. It is, in the deepest biological sense of the word, adaptive, not through deliberate strategy, but through the accumulated effects of countless tiny chemical decisions, each one made without awareness at the level of the individual hypha, each one contributing to the behavior of the living whole. Long before there was a word for it, long before there were laboratories or microscopes or the category of science we now call mycology, human beings were living in relationship with fungi. The relationship began, as most human relationships with other organisms begin, with hunger. Consider bread. The loaf that rises in a warm oven, that fills a kitchen with that particular yeasty warmth, that has been the center of tables across cultures for thousands of years. It is, in a very real sense, a collaboration between human beings and a single-celled fungus called Saccharomyces cerevisiae. Baker's yeast. An organism so domesticated by human use, so thoroughly embedded in human culinary culture, that it exists today in forms quite different from its wild ancestors, selected, over generations of human cultivation, for the properties that make it most useful for baking, the ability to consume sugars and produce carbon dioxide rapidly, inflating the elastic gluten network of wet dough and giving the resulting bread its characteristic open, airy texture. The carbon dioxide that leavens bread is a byproduct of the yeast's metabolism. The yeast is not trying to make bread rise. It is simply doing what living organisms do, consuming available energy sources and processing them in ways that sustain its own life. The bubbles of gas trapped in the dough are, from the yeast's perspective, waste products. From the human baker's perspective, they are the whole point. This relationship, in which humans harness the metabolic activity of a fungal organism to transform a raw ingredient into a food that would not otherwise exist, is replicated across dozens of different food traditions. Wine and beer fermentation depend on yeast converting sugars to alcohol. Sake is brewed with the help of a mold called Aspergillus oryzae, which produces enzymes that break down the starch in rice before yeasts convert the resulting sugars to alcohol. The dense, pungent, nutritionally rich flavor of many aged cheeses is partly the product of fungal activity, Penicillium roqueforti in blue cheeses. Penicillium camemberti in the white rind varieties, their metabolic processes transforming the proteins and fats of the cheese in ways that develop characteristic aromas and flavors over weeks and months of maturation. Soy sauce, miso, tempeh, fundamental proteins of Asian cuisines, are all produced through fungal fermentation. The transformation in each case is remarkable. Raw soybeans become, through the action of molds and yeasts and bacteria, foods of extraordinary complexity and nutritional depth. The fungi are not flavoring these foods. They are constructing them. The culinary relationship extends, of course, to mushrooms themselves, eaten directly, rather than as agents of transformation. Humans have gathered and eaten wild mushrooms for as long as there have been humans in forests. Truffles, chanterelles, porcini, morels, shiitake, oyster mushrooms, each with its own distinctive texture and flavor profile, each embedded in the culinary traditions of the cultures that have long lived alongside them. The flavors that mushrooms contribute to cooking are partly a result of their unusual biochemistry. They are rich in glutamates, compounds that activate the taste receptors responsible for what Japanese food scientists long ago named umami, the savory, full-mouthed, depth of flavor sensation that makes broths richer and sauces more satisfying. This glutamate richness makes mushrooms a natural flavor enhancer, and it partly explains why mushroom-based stocks and dried mushrooms have been used across many cultures as foundational flavoring ingredients. The human relationship with fungi extends beyond food. From the ancient world, various cultures have used fungi in contexts that range from the practical to the ceremonial, from medicinal preparations to spiritual practices. Documentation of these uses across cultures is extensive, though the specific effects of many traditional preparations remain subjects of research rather than settled understanding, and it would be misleading to draw simple lines between traditional use and modern pharmaceutical knowledge. What is documented and studied in detail is the story of how a chance observation in a London laboratory in the late 1920s led to one of the most consequential discoveries in modern medicine. Alexander Fleming, returning from a holiday, noticed that a petri dish of Staphylococcus bacteria had been contaminated by a mold, Mycelium notatum, and that the bacteria around the mold had died. Fleming recognized that the mold was producing something that killed the bacteria. He named the substance penicillin. The subsequent development of penicillin as a clinical antibiotic, the work of Howard Florey, Ernst Boris Chain, and many others through the 1930s and 1940s, transformed medicine in ways that are difficult to overstate. Bacterial infections that had been reliably fatal became, in the span of a few years, treatable. Surgeries that had previously carried enormous risk of post-operative infection became significantly safer. The discovery launched the age of antibiotics and initiated a decades-long search for other useful compounds in fungal and bacterial biochemistry. Today, that search continues. Fungi produce an extraordinary diversity of chemical compounds, many of them, from the fungus's perspective, likely involved in defense against competing organisms or environmental stresses. Some of these compounds have pharmacological properties of interest to medicine. Research is ongoing into various fungal metabolites, and the science in this area is active and evolving. The history of penicillin justifies continued attention to what fungi produce, while also serving as a reminder that the distance between laboratory discovery and clinical application is long, uncertain, and requires rigorous investigation. The relationship between humans and fungi, in all its dimensions, culinary, agricultural, industrial, medical, and more, is one of the oldest and most complex interspecies partnerships in human history. We have been fermenting with fungi, eating fungi, and benefiting from fungal activity in soil and decomposition for all of recorded history, and well before it. We are, in the most literal sense, embedded in a relationship with this kingdom that we are only now beginning to understand in its full depth. And what makes this relationship genuinely remarkable is not only its length or its intimacy, but its one-sidedness in terms of awareness. The fungi have no knowledge of us. They have been doing what they have always done, and we have happened to benefit. The gratitude, if it belongs anywhere, belongs on our side. The soft, quiet body of a mushroom, growing in the leaf litter of a forest floor, is without armor and without speed. It cannot run from what approaches it. It cannot strike back against what attacks it. And yet, across hundreds of millions of years of evolution, it has developed something that serves many of the same protective functions as armor and speed combined: chemistry. The chemical compounds produced by fungi represent one of the most diverse and structurally complex pharmacopoeias in the living world. Some are pigments that color the cap or gills. Some are volatile compounds that create distinctive aromas, attracting the insects that will carry spores or repelling organisms that might damage the fruiting body. Some are enzymes deployed outward into the substrate, breaking down complex molecules for nutrition. And some, the ones we tend to pay most attention to, are compounds that are biologically active in ways that can cause significant harm to organisms that attempt to consume the mushroom. We call these compounds toxins, though the word requires some care. Toxicity is not a fixed property. It is a relationship between a compound, a dose, and a recipient organism. A compound that is lethal to a beetle at microscopic concentrations might have no effect at all on a deer. A compound that affects a mouse profoundly might affect a human quite differently. The question of why certain mushrooms contain certain compounds is, at the most fundamental level, an evolutionary one, not a question about human experience at all. The mushroom is not, to state the obvious, trying to poison people. Humans are a remarkably recent addition to the mushroom's world. The compounds it produces have been shaped by selection pressures involving the organisms it has been interacting with for far longer: insects, bacteria, other fungi, and various animals that might eat it. When we ask why a mushroom produces a particular alkaloid or peptide that happens to be harmful to humans, the answer is almost always to be found not in any relationship with us, but in the ecological relationships that shaped the mushroom's chemistry long before any human was present to observe it. Some toxins appear to function primarily as defenses against insects. Certain compounds in mushroom tissue are effective deterrents against larval insects that might otherwise consume the fruiting body before its spores have been released. These compounds may have no significant effect on mammals or birds, which are not the primary threat the mushroom has evolved to resist. The evolutionary logic is not general deterrence but specific deterrence, targeted at the organisms most likely to cause harm at the most vulnerable moments. Other compounds may be more broadly active, not because the mushroom has evolved to deter many different organisms, but because the biochemical structures that evolved for one purpose happen to affect multiple types of biological systems. The molecules of life are, at their most fundamental level, shaped by chemistry, and chemistry does not respect the boundaries between species. Amatoxins, the compounds produced by certain Amanita species and some other mushrooms, represent one of the most studied examples of fungal defensive chemistry. These are cyclic peptides, small protein-like molecules whose structure gives them a particular and troublesome property. They inhibit an enzyme that is essential for the process by which cells read genetic information and make proteins. In organisms that consume them, this interference with basic cellular machinery can have severe consequences for tissues that are rapidly turning over cells, the liver and kidneys being particularly vulnerable. The evolutionary story of amatoxins is interesting precisely because it raises the question of who, originally, they were made to deter. Research suggests that amatoxins may have originated as compounds directed against bacteria or other microbial competitors rather than animals. Their effects on vertebrates may, in evolutionary terms, be incidental, a byproduct of chemical structures that evolved for entirely different reasons. Other classes of fungal compounds are better understood as competitive weapons between fungal species. Many molds and yeasts produce antibacterial compounds, including, of course, the penicillin produced by Mycelium molds, that suppress the growth of competing bacteria in their immediate environment. These are not toxins in the sense of being harmful to the fungus that produces them, but they function as chemical territories, staking out space for the fungus at the expense of its microbial neighbors. Some mushrooms produce compounds that affect the nervous systems of vertebrate animals. These may function as deterrents to grazing animals, organisms that might otherwise consume the entire fruiting body before spore release. The effect on the animal: disorientation, visual disturbance, behavioral changes, may be sufficient to discourage repeated consumption. The chemistry involved is varied and species-specific, and the mechanisms by which different compounds interact with neurological systems are complex and still being worked out in many cases. What is perhaps most striking about the defensive chemistry of fungi, viewed from a distance, is its sheer diversity. The molecular structures involved are not variations on a single theme. They are genuinely different solutions to the same broad problem: how to survive in a world full of organisms that might want to consume you. Each fungal lineage has, over evolutionary time, developed its own chemical solutions, its own suite of molecules shaped by the particular pressures of its own ecological history. This diversity is itself a kind of evolutionary achievement. The capacity to produce novel chemical structures, to explore chemical space in ways that other organisms cannot or have not, is part of what makes fungi such prolific sources of biologically active compounds. The same diversity that has given us compounds of medical interest has also given us the full spectrum of fungal toxicity, not as a threat or a curiosity, but as the ongoing result of billions of years of evolutionary experimentation in chemistry. The forest floor, seen from this angle, is not a passive landscape but a constant, slow chemical conversation, each organism producing and responding to the compounds of its neighbors, each chemical signal shaping behaviors and outcomes across the whole community. The mushroom's toxins are one voice in that conversation. A voice shaped not by any intention, but by the long patience of natural selection. If you were to design a condition hostile to life, to choose a set of parameters deliberately calculated to exclude as many organisms as possible, it would be difficult to find one that fungi have not, somewhere, managed to survive. This is not hyperbole. It is a reflection of the extraordinary range of environments in which fungal organisms have been found, thriving, reproducing, and doing the slow biochemical work that fungi do, in conditions that most life would find intolerable. Consider the world's deserts. These are environments characterized by extraordinary heat during daylight hours, dramatic temperature swings between day and night, intense ultraviolet radiation, and, crucially, profound and extended dryness. Most life requires water to function. Metabolic processes depend on water as a solvent, as a medium for chemical reactions, as a carrier of nutrients. Prolonged desiccation is, for most organisms, lethal. And yet, in the crust of desert soils, in the biological soil crusts that form thin, stable films across the surface of arid environments, fungal hyphae are present. Certain fungi have evolved the capacity to tolerate water activities so low that most bacteria could not survive. They can enter dormancy during the driest periods, and resume metabolic activity when even small amounts of moisture become available, from morning dew, from occasional rain, from the microscopic capillary water films that persist between soil particles even in apparently dry conditions. Some of these desert fungi are melanized, their cell walls darkened by melanin pigments that may provide protection against the intense ultraviolet radiation at the soil surface. Melanin absorbs UV light and may also help these organisms manage oxidative stress. The same pigments that give certain fungi their dark, sometimes almost black appearance in extreme environments, may be, in part, their shield against the radiation that would otherwise damage their DNA. The ocean floors present a very different set of challenges: cold, darkness, pressure. At depths of hundreds or thousands of meters, the pressure of overlying water is enormous, temperature approaches zero, sunlight is entirely absent, and the chemical environment is profoundly different from that of terrestrial soil. And yet fungal organisms have been recovered from deep-sea sediments, from pressures that would compress most organisms beyond function, in darkness that makes photosynthesis impossible, in environments so remote that their existence was long unsuspected. The fungi found in deep-sea environments appear to include both species related to terrestrial lineages, organisms that may have been transported to the ocean floor over time in settling organic material, and potentially novel species adapted specifically to the deep-sea environment. Research in this area is still at an early stage, and the full diversity and ecological role of deep-sea fungi is only beginning to be characterized. At the other extreme of temperature, fungi have been found in the polar regions, in the soils of Antarctica, beneath the ice of glaciers, in the frozen sediments of permafrost. Some cold-adapted fungi, psychrophilic species, have optimum growth temperatures well below what most organisms would consider comfortable, and some can continue metabolic activity at temperatures near or even slightly below the freezing point of water, in thin films of liquid that persist between ice crystals and frozen soil. On rocks, in patterns that look more like staining than living tissue, endolithic fungi grow within the pores and crevices of stone, sometimes centimeters below the rock surface, where temperature and moisture fluctuations are buffered by the surrounding mineral matrix. These organisms extract what nutrition they can from the rock itself, dissolving minerals through acid secretion and absorbing the resulting ions. They contribute, very slowly, to the biological weathering of rock, the gradual conversion of mineral matter to soil that underlies the formation of terrestrial ecosystems. Among the most remarkable of the extreme fungal lifestyles are those involving intimate associations with insects. Certain fungi have evolved to colonize living insects, not as opportunistic infections of weakened or dying individuals, but as highly specific, co-evolved relationships with particular host species. The cordyceps fungi, which belong to the Ophiocordyceps genus and related groups, are the most famous examples. These organisms infect specific insects, in some species, specific life stages of specific insects, with a precision that suggests long evolutionary calibration. The fungal spore lands on the exoskeleton of the appropriate host insect and germinates, with hyphae penetrating the cuticle and entering the body cavity. There, the fungus grows within the insect, ultimately killing it. In some species, the behavior of the infected insect changes prior to death in ways that bring the insect to a location favorable for spore dispersal, climbing vegetation, for example, in a way that places the dying insect above the forest floor where other insects of the same species forage. It is important to be precise about what this means, and what it does not mean. The fungus is not controlling the insect in any intentional sense. Rather, the fungal infection produces chemical compounds, some of which may interact with the insect's nervous system, that alter the insect's behavior in ways that happen to benefit the fungus's reproductive success. How the specific behavioral modifications are achieved, and exactly which compounds are responsible, is an area of active research. The results are genuinely extraordinary from a biological standpoint, without requiring any element of intention or agency on the part of the fungus. The diversity of these extreme habitats suggests something important about the nature of fungal organisms. They are, perhaps more than any other large kingdom of life, environmental generalists in the deepest sense. Their absorptive mode of nutrition allows them to exploit chemical energy wherever it is found. Their capacity for dormancy allows them to wade through conditions that would kill actively metabolizing organisms. Their dispersal by spores means that wherever conditions are even marginally suitable, there is a chance that fungal life will eventually arrive and establish. The Earth has, over its history, offered an enormous variety of environments, and fungi have explored nearly all of them. This breadth of ecological range is not a coincidence. It is the outcome of an evolutionary experiment that has been running, with modifications, for something close to 1 billion years. There is a forest in the Blue Mountains of eastern Oregon, in the United States, where a single organism is growing. You would not know it by looking. The forest appears, to any eye, to be a diverse community of trees, a mix of conifers and smaller plants, light filtering through canopy, the ordinary appearance of a healthy temperate wood. But beneath the surface of that forest, spreading through the soil across what researchers estimate to be an area of perhaps many hundreds of hectares, is a single continuous mycelial network of Armillaria ostoei, commonly known as the honey fungus. The mushrooms that this network occasionally produces, small, honey-colored caps clustered at the bases of trees, are the only visible indicators of its presence. And the mushrooms, as we now know, are only the fruiting bodies, temporary expressions of the organism below. The organism itself is the mycelium, and the mycelium of this particular individual is, by some measures, the largest single organism known on Earth. The qualification is important to hold carefully. Estimates of its age, sometimes cited as potentially thousands of years old, are based on models of how quickly the mycelium might have expanded from a single point of origin, combined with genetic evidence that the cells across the network share the same genetic identity. These are estimates, not certainties. The precise age of any such organism is extremely difficult to determine directly, and researchers are appropriately cautious about the numbers they attach to it. What does seem well supported is that this is an individual organism, a single genetic entity, of remarkable extent. The Armillaria genus contains organisms known to produce very large, expansive mycelial networks. The growth pattern of these fungi involves sending out rhizomorphs, thick, root-like strands of compressed mycelium that extend through the soil more rapidly than individual hyphae and allow the network to cover large areas over time. The fungal organism grows not simply as a diffuse cloud of hyphae, but as a structured, spreading network with channels for transport and regions of active growth. The question of what it means to be a single organism, at the scale of a large fungal network, is philosophically interesting. The cells at opposite ends of such a network may be hundreds of meters apart, separated by conditions that vary significantly, different soil chemistry, different moisture levels, different temperature regimes. In the ordinary sense, each small portion of the mycelium is living its own local life, responding to its immediate environment, growing or pausing according to local conditions. And yet, genetically, all of these cells share the same heritable information. They are the product of the same original event, the germination of a single spore, and the expansion, over time, of the mycelium that germinated from it. This raises questions about the nature of individuality in organisms that grow indeterminately, and that do not have a body plan in the conventional sense. A human is an individual because, among other reasons, all the cells in the human body share genetic information and are derived from a single founding cell. By this criterion, the large Armillaria network is also an individual, but it lacks the other markers we typically associate with individuality. It has no defined boundary, no self-contained metabolism, no nervous system that integrates information across the whole. Some mycologists prefer to speak of genets, genetic individuals, to distinguish this form of individuality from the physiological individuality of animals. A genet is simply a collection of cells that share the same genetic origin, regardless of how spatially dispersed or metabolically integrated they are. The large Armillaria in Oregon is, by this definition, a single genet, one genetic individual, occupying an enormous space. The Armillaria case is the most extensively studied example of a very large fungal individual, but it is not unique. Other fungal networks of great extent have been documented in various parts of the world. Research techniques based on genetic analysis, the ability to determine whether cells from different locations in a forest share the same genetic identity, have allowed scientists to begin mapping the extensive individual fungal organisms in ways that were previously impossible. What these studies consistently reveal is that the spatial scale of fungal individuals can be dramatically larger than the scale of the mushrooms they produce, and that the assumption of many distinct fungal individuals in a forest may sometimes be an underestimate of the actual degree of genetic continuity in the underground network. There is something genuinely moving about this, once you sit with it for a moment. An organism whose presence you might walk through without suspecting. An individual that has been growing for longer than most of the trees around it have been alive, spreading its network through the soil of a forest in an expansion so gradual it has no perceptible motion, occupying space on a scale that no single animal can match, doing all of this without hurry, without drama, without any visible announcement of its magnitude. The patience of the mycelium, the long, quiet accumulation of presence over time. It is one of the stranger portraits of biological success that the natural world has to offer. Beneath your feet, in almost any vegetated environment on Earth, carbon is stored. It is stored in the form of organic molecules, the bodies of living organisms, the partially decayed remains of organisms that have died, the stable humus compounds that form when organic matter has been processed by soil life many times over. The amount of carbon stored in soils globally is estimated to be substantially greater than the amount currently in the atmosphere, and larger than the amount stored in all living vegetation on the surface. Soil carbon is, in other words, a significant reservoir in the global carbon cycle, one that interacts continuously with the atmosphere and plays an important role in the Earth's long-term climate regulation. Fungi are deeply involved in the dynamics of this reservoir. At the most basic level, decomposer fungi release carbon from dead organic matter. When a fungus breaks down a fallen tree or a layer of accumulated leaf litter, the carbon in those materials is processed. Some of it is consumed by the fungus and respired as carbon dioxide, returned to the atmosphere. Some of it is incorporated into the fungal biomass itself, and some of it, particularly the more resistant carbon compounds, is left behind in forms that may persist in the soil for long periods. This partitioning of carbon between different fates, respired to atmosphere, incorporated into living biomass, or stabilized in the soil, is not a trivial detail. It is a key variable in determining how much carbon a given ecosystem retains over time. Fungi that leave large amounts of stabilized organic carbon in the soil, either through incomplete decomposition or through the production of fungal compounds that are themselves resistant to further breakdown, contribute to the long-term storage of carbon in the ecosystem. Some research suggests that ectomycorrhizal fungi, in particular, may contribute to the formation of stable soil organic matter in ways that differ from the contributions of saprotrophic fungi. The mechanisms are complex and still being studied, and the specific magnitude of these contributions varies between ecosystems and fungal communities. But the broad picture that emerges from this body of research is one in which fungi are not passive elements of the carbon cycle, but active participants, organisms whose metabolic activities and the compounds they produce influence how carbon moves through ecosystems. Mycorrhizal fungi also represent a significant pool of living carbon themselves. The biomass of fungal mycelium in soils is not trivial. Research has attempted to estimate the total fungal biomass in various ecosystems, and while precise global estimates involve considerable uncertainty, the numbers suggest that fungal biomass in soils is substantial enough to matter in ecosystem-scale carbon accounting. The interaction between fungi and climate also operates in the other direction. Fungal communities respond to changes in temperature, moisture, and atmospheric chemistry. Research suggests that the composition of fungal communities in soils, which species are present, and at what abundances, is sensitive to environmental conditions. As those conditions shift, fungal communities may change in ways that alter the rates and products of decomposition and nutrient cycling. Warming temperatures, for example, may accelerate the metabolic activity of soil fungi in some environments, potentially increasing the rate at which carbon from soil organic matter is converted to carbon dioxide. In other environments, changes in moisture regimes may be equally, or more, important. Drought can suppress fungal activity, reducing decomposition rates and potentially leading to greater carbon storage. Excess moisture can do the opposite in some systems. These interactions between fungal communities and climate variables are genuinely complex, and researchers are cautious about drawing simple conclusions. The feedback loops between soil biology and climate are multidirectional and difficult to model with precision. What seems clear is that soil fungal communities are not merely passive bystanders in climate dynamics, but active participants in the processes that regulate how much carbon is stored in the landscape and how much circulates in the atmosphere. This understanding carries implications for how we manage landscapes, forests in particular. Soil fungal communities can be disrupted by land management practices, intensive tillage, the use of certain pesticides and fertilizers, clear-cutting of forests, the conversion of natural vegetation to agricultural use. When these communities are disrupted, the processes they mediate, decomposition, nutrient cycling, carbon stabilization, are also disrupted. The recovery of fungal communities after such disturbances can take considerable time, and the trajectories of recovery are not always predictable. The picture that emerges, when you put these pieces together, is of fungi as one of the most consequential organisms on Earth for the functioning of the global carbon cycle, not through any dramatic action, but through the slow, constant, distributed activity of trillions of organisms, each doing the work that fungi do, each contributing incrementally to the outcomes that matter at the scale of the whole planet. The forest floor is not just scenery. It is a climate system. There is a particular kind of patience required to watch a mushroom grow. Not the patience of watching a flower open, which can be perceived in real time, the petals uncurling in the warmth of a sunny morning. Mushroom growth is partly too slow, and partly too fast, for comfortable observation. The early stages of development take place underground, in darkness, over hours or days. The later stages, the dramatic expansion from small, rounded primordium to fully open cap, can happen with surprising speed, in a matter of hours, while most observers are sleeping. The story begins long before any mushroom is visible above the surface. It begins in the mycelium, when conditions have aligned in a way that triggers the organism to redirect its resources toward reproduction. What constitutes the right conditions varies between species, but temperature, moisture, and the chemistry of the surrounding environment all play roles. Many mushrooms are triggered by a drop in temperature following a warm period, the conditions that occur in autumn in temperate regions, when summer warmth gives way to cooler nights and the first autumn rains arrive. The mycelium, which has been extending through the soil and processing organic matter through the warmer months, responds to this shift by beginning to organize certain of its hyphae into a new kind of structure. The first visible stage of this organization is called a hyphal knot, a small, dense aggregation of hyphae in which individual threads are growing more compactly and branching more frequently than in the surrounding network. A hyphal knot is not yet a mushroom, or even a recognizable precursor to one. It is the mycelium beginning to make a decision to concentrate energy and material into a particular location, to build something rather than simply extending outward. If the knot is sustained, if conditions remain suitable, and the mycelium continues to supply materials to the developing structure, it gradually develops into what mycologists call a primordium. This is the first recognizable precursor of the fruiting body, a small, often rounded or button-like structure that contains, in compressed and folded form, all the essential architecture of the mature mushroom: the cap, the gills, the stem, packed tightly together and waiting for the signal to expand. The primordium is often visible to the naked eye, though small, perhaps a few millimeters in diameter, sometimes less. If you were to section it carefully and examine it under magnification, you would see the layered arrangement of tissues that will eventually become the distinct parts of the mushroom. The gill tissue is already there, in folded, compressed form. The cap tissue is there, folded around it like a closed hand. Everything is present. Nothing has yet expanded. Expansion, when it comes, is primarily a story of water. The cells of the developing mushroom take up water rapidly, drawn in through the hyphal connections from the surrounding soil, and transported upward through the developing stem in a hydraulic movement that requires no pumping, only the osmotic gradients created by the accumulation of compounds within the mushroom cells. As water floods into the cells, they swell. The tissues of the cap straighten as the gill surface unfolds from its compressed configuration. The stem elongates. The cap rises above the soil on an increasingly long stalk. This expansion is rapid enough, in some species, to be perceptible on camera at normal speed over the course of a few hours. The time-lapse films of mushroom growth that circulate widely online, showing caps unfolding from tight buttons to fully open structures in seconds, are genuinely representative of the actual speed of late-stage development, only slightly compressed in time. A cap that looks tiny and rounded in the morning may be fully expanded by evening. The geometry of the expanding cap is not arbitrary. The cap surface grows in a pattern that results, in most species, in a roughly circular form that maximizes the area of gill tissue exposed to the air beneath it. The gills themselves are arranged in precise radial patterns, like the spokes of a wheel, or the pleats of a folded fabric, in configurations that vary characteristically between species. This variation in gill arrangement is partly the basis on which taxonomists distinguish between fungal species, and it reflects genuine differences in the architecture of the tissue that produces and releases spores. As the cap expands and the gills open, spore production begins on the gill surfaces. Each gill surface is lined with cells called basidia, spore-producing cells that undergo cell division and produce 4 spores apiece, each one attached to a small sterigma that will, at the appropriate moment, use the Buller's drop mechanism to launch the spore into the airspace between adjacent gills. The mature mushroom, fully expanded, is a precisely calibrated spore dispersal device. The cap is angled to direct falling spores downward through the gill spaces. The cap's elevation above the substrate ensures that spores, once launched, enter air currents that can carry them away from the parent organism. The entire architecture of the fruiting body, the relationship between cap, gill, stem height, and pore geometry, can be understood as an integrated solution to the problem of getting spores from inside the gill surface into the wider world. And then, having solved that problem, having released its spores into the waiting air over hours or days, the mushroom's work is done. Water is no longer being supplied from the mycelium below. The cells of the cap begin to collapse as their internal pressure drops. The soft tissue softens further, darkens, begins to dissolve. The cap droops and folds. The gills drip. Within days, sometimes within hours, what was a crisp and structured object has become a soft, dark, liquefying thing that will be absorbed back into the soil. The mycelium continues. The cycle restarts. A mushroom cap, viewed from below, is a map of geometry. The gills, those thin, closely spaced vertical plates radiating outward from the stem, are arranged in precise patterns that vary characteristically between species. In some mushrooms, every gill runs all the way from the stem to the outer edge of the cap. In others, shorter gills alternate with the full-length ones. In some species, the gill margins are serrated. In others, They run smoothly. Some gills are wavy. Some are forked near the cap edge. The specific pattern, seen at the right angle and in the right light, has the quality of a careful architectural drawing, regular, purposeful, aesthetically compelling. These patterns are not decorative. They are functional solutions to a geometric problem: how to pack the maximum area of spore-producing surface into the space available beneath a mushroom cap. A flat surface produces spores from one side only. Folded into gills, the same area of tissue can produce spores from both sides of each fold, while also creating the air spaces between gills necessary for spore dispersal. The narrower the gill spacing and the deeper the gills, the greater the total spore-producing surface area per unit of cap area. But spacing cannot be reduced without limit. If gills are too close together, the air spaces between them become so narrow that spores cannot be effectively launched and dispersed. The gill arrangement in any given species represents, in this sense, an evolutionary optimization, a configuration that balances the competing demands of maximum spore production area and adequate airflow. Different lineages of fungi have arrived at somewhat different solutions to this optimization, which is partly why gill patterns vary so consistently between groups. Some fungi have abandoned gills entirely in favor of different spore dispersal architectures. Boletes, the group that includes the porcini and many other familiar edible mushrooms, have pores instead of gills, vertical tubes packed closely together on the underside of the cap, each tube producing spores along its inner wall. The tube architecture achieves a similar increase in spore-producing surface area relative to a flat surface, through a different geometric strategy. Chanterelles have forked ridges, structures that are sometimes described as false gills, that create a textured spore-producing surface without the discrete, plate-like organization of true gills. Bracket fungi, growing on the sides of trees, often have pore surfaces and shape their caps not as horizontal platforms but as angled shelves, with the pore surface directed downward regardless of the orientation of the supporting substrate. Some species adjust the angle of cap growth in response to gravity, ensuring that spores will fall through the pore openings in the right direction even when the substrate on which the fungus is growing is not vertical. This gravitropic response, growth in response to gravity, is a direct expression of the functional requirement for vertical spore fall. At a smaller scale, the branching patterns of hyphae also reflect geometric principles. Hyphal branching in many species follows patterns that have been described mathematically as fractal-like, patterns that exhibit self-similarity at different scales, in which the branching structure of the whole mycelium resembles the branching structure of each individual hypha, which in turn resembles the branching at subhyphal scales. This kind of self-similar branching is characteristic of many natural growth processes, and emerges from simple, locally applied rules of growth, rules like branch when you encounter resistance or branch at a certain frequency of extension, that produce complex, space-filling patterns without any global plan. The fractal character of mycelial networks is not incidental. It is, again, a solution to a geometric problem: how to explore a three-dimensional space most thoroughly with the minimum amount of material. A highly branched, space-filling network maximizes contact between the fungal organism and the substrate through which it is growing, maximizing the surface area available for enzyme secretion and nutrient absorption relative to the volume of fungal material produced. These geometric principles, the optimization of surface area, the space-filling properties of fractal branching, the directional growth in response to gravity, are not unique to fungi. They appear across the living world, in the branching of trees, the folding of intestinal surfaces, the architecture of lungs and circulatory systems. Life has, independently and repeatedly, discovered that certain geometric arrangements are solutions to certain functional problems. The mushroom's gill pattern and the mycelium's branching pattern are simply the fungal expression of these universally useful geometries. There is something satisfying, in a deep and quiet way, about recognizing this. The patterns we find beautiful in the underside of a mushroom cap, the regularity, the repetition, the fine-scaled elaboration of a simple motif, are not separate from the pattern's function. They are the same thing. The beauty and the purpose arise together, from the same geometric logic, shaped by the same evolutionary pressures, discovered by the same long process of trial and modification. When you hold a mushroom and turn it over in your hands, you are holding not just an organism, but a solution to a problem, one that took hundreds of millions of years to refine. Let us pause here, for a moment, and not rush onward into the next piece of knowledge. There is something worth sitting with, a sensation that arises when you have been thinking carefully about fungal life for some time, and begin to feel the weight of what is invisible. Not the specific facts, though the facts are remarkable. Something else. Something more like a shift in the texture of how the world seems. We spend most of our lives navigating the visible. We trust what we can see, what we can touch, what announces its presence through light and sound and direct sensation. The invisible is, in most ordinary contexts, something to be suspicious of, something that science has been designed, in part, to render visible through instruments and measurement and careful inference. And there is wisdom in this. The visible world is real, and it repays attention. But the visible world, it turns out, is only part of the world. Beneath every field, beneath every garden, beneath the parking lots and the pavements and the lawns and the managed forests of the world we have built, there is a living system of extraordinary complexity operating beyond the reach of our eyes. Mycelium threading through soil particles. Networks connecting roots to fungi and fungi to each other. Decomposer hyphae processing the fallen and the dead. Spores waiting in dormancy for conditions that may not arrive for years. Chemical signals moving through water films between organisms that have been exchanging them for millions of years. We did not build this system. We did not design it. It was here long before we were, and in most places, it continues its work without any knowledge of our presence or any relationship with our activities beyond the incidental ones, the way our footsteps compress soil, the way our buildings displace the organisms that would otherwise live there. What does it mean, to live above a world you cannot see? The question is not only about fungi. It is about the general condition of being a creature with limited senses in a reality of vast complexity. We see a narrow band of the electromagnetic spectrum. We hear within a particular frequency range. We detect chemistry through smell and taste at sensitivity thresholds that exclude the vast majority of the molecular information present in our environment at any moment. We navigate time at a scale that makes us nearly blind to both the very slow and the very fast. We are partial observers of a reality that exceeds our observation in every direction. And yet, we are not therefore ignorant. We have developed tools, microscopes and mass spectrometers and DNA sequencers and mathematical models that extend our perception beyond the limits of our unaided senses. We have developed methods of careful inference that allow us to construct reliable knowledge about things we cannot directly observe. We have, in short, learned to make the invisible at least partially visible, through the patient application of intelligence and method. Mycology is, in many respects, the science of learning to see what was always there but was not being looked at. The mycelium was always beneath our feet. The spores were always in the air we breathed. The fungal-plant partnerships were always operating in the roots of every tree we walked beneath. What changed was not the fungi. What changed was us, our curiosity, our instruments, our willingness to direct sustained attention towards something small and dark and underground, and ask what it was doing. There is a kind of humility that this demands, and that perhaps this particular moment in history, with its easy access to images and data and the accumulated knowledge of science, makes harder to practice than it would have been in other eras. We are surrounded by information, and information can create the feeling of comprehensiveness, the sense that what is known is the majority of what there is to know, that the maps are mostly filled in, that the age of discovery is mostly complete. Fungi suggest otherwise. Estimates of the true number of fungal species on Earth, many millions, perhaps, by some calculations, compared with the approximately 150,000 currently described, suggest that the majority of fungal life remains unknown to science. The ecology of soil fungal communities, how they structure themselves, how they respond to disturbance, what drives their composition in different environments, is a field in which major questions are still open. The biochemistry of fungal compounds, the evolutionary history of the many branches of the kingdom, the interactions between fungal communities and climate, between fungi and the other members of the soil food web, each of these areas contains more open questions than settled answers. The hidden world beneath us is not only physically hidden, it is intellectually hidden, incompletely described, only partially understood, still yielding its secrets slowly to the accumulation of scientific attention. Perhaps that is one of the most important things that fungi have to offer us, beyond bread and decomposition and the minerals they release into soil, a reminder of the scale of what remains unknown. A reminder that the world is larger than our current model of it, deeper than our current instruments can reach, more complex than our current frameworks can fully capture. Not in a mystical sense, in a straightforwardly empirical sense. There is simply more to find out. And if a forest depends on invisible threads, if the health of every tree is partially a function of organisms too small to see, operating in processes too slow to observe directly, then the question naturally arises, where else in life do hidden systems carry the weight? Where else are the structures that matter most the ones that are least apparent? Where else is the surface a consequence of the depth? These are not rhetorical questions. They are genuinely open ones, in ecology and in the broader consideration of how complex systems function. The mycelium is one answer to the question of what hidden things can look like. There are others. And the practice of looking, carefully, patiently, with instruments when instruments are needed and without presumption about what one will find, is perhaps the most important methodological inheritance that the study of fungi can offer. Slow down. Look more carefully. There is more here than is apparent. There is a quality of excitement in contemporary mycology that feels, from the outside, like the excitement of a field that has recently discovered it is much larger than it thought. For much of its history, mycology, the scientific study of fungi, was a relatively quiet discipline. Important, certainly, but not glamorous. Fungi were understood primarily in terms of their agricultural relevance, as pathogens of crops, as producers of useful compounds, as the organisms responsible for the decomposition of organic matter. The ecological dimensions of fungal life, the symbiotic networks, the diversity of soil communities, the global scale of fungal activity in carbon and nutrient cycles, were known in outline, but not in detail. What changed, in the last few decades, is primarily the availability of powerful tools for genetic analysis. DNA sequencing technologies that once required months of laboratory work and substantial cost can now be applied rapidly and cheaply to environmental samples, a handful of soil, a section of tree bark, a sample of sediment from the ocean floor. These methods have allowed researchers to begin characterizing fungal communities without needing to grow the organisms in culture, a significant constraint that had previously limited our understanding. because the vast majority of fungal species in any environment cannot be easily cultivated in the laboratory. The results of this genomic revolution in mycology have been, by any measure, extraordinary. Every study of environmental DNA from soils, every careful analysis of the fungal genetic material present in samples from forests, agricultural fields, grasslands, deserts, reveals a diversity of fungal species that substantially exceeds what was previously catalogued. New lineages, new relationships between known groups, species unknown to science, in environments assumed to be well studied. This expanding sense of what fungi are, and what they do, has opened new areas of practical application, areas being explored with genuine enthusiasm and appropriate scientific caution. Bioremediation, the use of living organisms to clean up contaminated environments, has attracted considerable interest in the context of fungi. Certain fungi produce enzymes capable of breaking down not only natural complex polymers like lignin, but also synthetic compounds, including some industrial pollutants that resist degradation by other means. The white rot fungi, with their battery of powerful oxidative enzymes, can in laboratory and controlled settings degrade a range of compounds of concern, including certain petroleum products and some persistent organic pollutants. The application of this biochemical capacity in real contaminated environments is more complex than laboratory experiments suggest. Soil is not a controlled environment. Competition from other organisms, variability in moisture and temperature, the need to distribute fungi effectively through three-dimensional soil, all of these factors complicate the translation from lab to field. Research in bioremediation continues, with careful attention to what works reliably under real-world conditions, as distinct from what works elegantly in a petri dish. Materials science represents another area of active exploration. The compressive strength and water resistance of fungal mycelium, combined with its ability to bind together and colonize organic substrates like agricultural waste, has led to the development of mycelium-based materials, blocks of bound, dried mycelium that can substitute for synthetic foams in packaging and insulation applications. Several companies have brought such products to commercial scale, and research continues into the properties of different fungal species on different substrates, with an eye toward expanding the range of applications. These materials have the notable property of being biodegradable. They will be decomposed, in an appropriate environment, by the same fungal and bacterial processes that decompose any organic matter. This places them in a different environmental category from the synthetic materials they may replace. Whether mycelium-based materials will prove competitive with alternatives across the full range of performance and cost considerations remains to be determined, but the research trajectory is active, and the early results are encouraging. Medical research involving fungal compounds continues to be an area of attention, building on the history of penicillin and the many other clinically useful compounds that have been derived from fungal sources. Research is ongoing into various classes of fungal metabolites, compounds that show activity against bacteria, fungi, parasites, or cancer cells in laboratory settings. The pipeline from laboratory activity to clinical treatment is long and uncertain, involving rigorous testing of safety and efficacy before any compound reaches patients. The history of medical mycology counsels both optimism about what fungi might yield, and patience about the timeline and probability of any given discovery translating into a treatment. There is also growing interest in the agricultural applications of mycological knowledge, particularly in the potential for mycorrhizal inoculants to improve the nutrient efficiency of crops, reducing dependence on synthetic fertilizers. The research here is active, and the concept is biologically sound, but the practical application involves significant complexity. Different crops partner with different fungal species, and the effectiveness of inoculants depends on soil conditions and the existing fungal community. Careful, Context-specific research is needed to identify where and how mycorrhizal management can reliably benefit agricultural systems. Underlying all of these practical applications is a more fundamental aspiration to understand fungal biology well enough to work with it intelligently. The history of human interactions with other organisms suggests that intelligence in this context means not just technical understanding, but also ecological humility, an appreciation for the complexity of the systems involved, and the ways in which interventions in those systems can have consequences that simple models do not predict. Fungi are not tools, in the simple sense of objects we can pick up and direct according to our purposes. They are organisms embedded in webs of relationships that we are still learning to describe. Any practical application that involves fungi will involve, to some degree, working within those relationships, understanding them well enough not to disrupt more than is intended. This is the most important disposition that mycology, and perhaps ecology more broadly, recommends, not control, but careful, curious, humble collaboration with the world as it actually is. The fungi will continue doing what they do, as they have for hundreds of millions of years, long after any particular human application has come and gone. Our task is to learn enough about their work, to benefit from it, without disrupting it. That learning has barely begun. The forest at night is different from the forest in daylight, though the trees are the same trees, and the soil is the same soil, and the mycelium threading through the dark beneath you has not changed its nature since the sun went down. What changes is what you can see, and what you can hear. And, in the absence of those familiar inputs, perhaps also what you can feel, or almost feel, in the quality of attention that the darkness asks of you. The mist that gathered in the morning has returned. It moves low through the trees, cooling the air where it touches, settling on every surface it encounters, on the smooth upper surfaces of fallen leaves, on the rough bark of old trunks, on the fine membranes of mushroom caps that stand pale and quiet in the understory. A moth moves between the trees, and all somewhere in the canopy adjusts its position. Otherwise, the forest is still. Beneath you, without drama, without announcement, without any sign that you could directly perceive, the work continues. The mycelium does not rest at night. It has no day-night cycle in the sense that you do, no circadian rhythm that draws it towards sleep and away from activity in the dark hours. It extends continuously, as it has been extending, tips probing forward through the compressed dark of the soil, enzymes flowing outward ahead of the growing point, nutrients streaming back along the hyphal channels toward the parts of the network where growth is most active. The fallen log at the edge of the clearing is, tonight as it is every night, being slowly dissolved by the patient work of hyphae that first colonized it perhaps a decade ago, and have been at work within it ever since. The roots of the oak above you, that great, still, winter bare oak, are connected, at their finest tips, to fungal networks whose hyphae extend far beyond the root zone in every direction. Through those connections, whatever is available in the soil, phosphorus, nitrogen, perhaps sugars moving in the other direction, is being exchanged in the slow, chemical language of mycorrhizal partnership. The oak does not know this, in any sense we would recognize as knowing. The fungi do not know it either. And yet the exchange proceeds, as it has for perhaps a century with this particular tree and this particular community of fungal partners, and it will proceed tomorrow, and through the winter, and into the spring when the tree's new leaves will begin their photosynthetic work again, and the whole cycle of carbon and mineral exchange will resume its warmer season pace. Somewhere in the leaf litter nearby, a spore landed today. It may have traveled from a forest miles away, carried in the slow autumn air. It has settled, for now, among the compressed leaves and the soil particles and the already present community of fungal life that occupies this square meter of forest floor. Whether it will germinate depends on what tomorrow brings, whether the moisture is sufficient, whether the temperature falls within its tolerance, whether the chemistry of this particular piece of soil matches what this particular spore was evolved to encounter. Most spores do not succeed. This one may or may not. It waits. There is something about the night that makes it easier to hold these thoughts. In the daylight, the world tends to assert itself visually. The visible parts of things press forward, demand attention, offer themselves as the reality. But in the dark, the visible retreats, and perhaps the invisible becomes a little easier to contemplate. The vast underground network is no more real at night than it is in daylight. But the dark makes it slightly easier to believe in. You cannot see the mycelium beneath your feet. You cannot hear it, or touch it, or smell it in any distinct way, though the earthy, complex smell of forest soil is partly a product of fungal activity, the particular volatile compound called geosmin that bacteria and fungi produce in the course of their metabolic work, and that the human nose is extraordinarily sensitive to. You cannot perceive, directly, any of the processes we have spent these hours following: the enzymatic dissolution of wood, the hyphal extension through soil, the exchange of phosphorus for sugar at the mycorrhizal interface, the launching of spores from gill surfaces. And yet, knowing it is happening, having followed it in thought through the patient accumulation of what science has learned, changes something. The soil beneath you has weight. It has history. It has what might almost be called a kind of life of its own, not metaphorically, but literally, a living system of staggering complexity, operating at scales of time and space that dwarf any individual observation. Most of life is not loud. It does not bloom for applause. It does not announce itself, or seek recognition. It does not hurry. The greatest networks on Earth extend themselves by the increment of a single hyphal tib, one growing point at a time, patient and continuous and unremarked. The most essential recycling of nutrients on land is accomplished by organisms invisible to the naked eye, working in darkness, in soil, at temperatures that most of us would call cold. The partnerships that sustain the trees, that sustain the atmosphere, that sustain us, are conducted in a chemical language so quiet and so slow that centuries of human observation failed to detect them. This is not a reason for despair. It is a reason for a different kind of attention, one that is slower than urgency, more patient than fascination, more willing than usual to sit with what cannot immediately be seen, or demonstrated, or resolved. Beneath your feet tonight, an entire kingdom is breathing. It has been breathing since before the first forests, since before the first insects, since before the first footprint on dry land. It will be breathing long after every visible thing above it has had its turn in the light, and returned to the ground from which it came. It does not need your knowledge of it to do its work. But perhaps your knowledge of it changes, in some small way, how you walk through the world. Go quietly. The soil is alive. The dark is full. Somewhere beneath a fallen leaf, a hypha extends its tip into the night. And the kingdom breathes.